Awesome. Thank you so much for that introduction. And thank you all for inviting me to be here. I feel like this is a really interesting audience for me to present to. I've spent my entire career focused on satellite DNAs, which have been kind of hidden away in the gaps of our genome, and waiting for a moment for a community like this to start paying attention to some of these repetitive sequences that haven't really been in our view until now. So let me see if I can change it. Yes. Wonderful. So I'm a genome geek, and I think all of us can recognize that the human reference genome has just been foundational for all of our genetic and genomic questions. And I'm showing this slide because I don't want you to think about the genome as being a string of A, T, C, Gs, but rather a coordinate system, similar to what we think about with Google Maps, where we have shared this coordinate system around the world for 2 decades. And what we've been doing as a community is that we've been adding information to this coordinate-based system, information about sequences that are different between individuals around the world or population genetics, information about basic understanding about how the genome functions, and also about clinical genetics and genomics. And the reason why that's so important is because I want you to understand that for the past 2 decades, roughly 10% of this map was incomplete, and we've known it. We celebrated in 2003 the release of the finished human reference genome, but that was really 99% of the human euchromatic portions. And the regions that we knew were heterochromatic. These are regions that are found in the subtelomeric regions, the entire acrocentric short arms, centromeric regions, and even included duplicated gene families within the arms were known to be missing. And I want to use this slide to really emphasize that these regions are important for life. If you were handed a genome without these gapped regions, the cell would simply not live. It's because we know that the regions that were missing represent centromeric sequences, are important for how the cell has a chromosome transmission every single time it divides faithfully. We know the rDNAs are important for understanding a protein translation. And of course, these gene families can help us to really understand not only the mechanisms of dosage of having different numbers, but also their involvement in genome instability. And I really always put this up because as much as I'm going to tell you today, there's decades more that we don't understand. What I'm showing you is not a human cell. This is a mouse cell that looks like a chocolate chip cookie. But what I'm trying to emphasize are these bright or darkly staining chromocenters, which represent some of these newly introduced sequences that we know are highly repetitive. And we know these regions of our genome are not just topping up. This is not just adding more A, T, Cs, and Gs. These are regions that have been completely overlooked in terms of understanding chromosome structure and function. We know they're late replicating. We know they have specialized DNA repair, chromatin organization, and the list goes on and on and on. And so what I'm here to talk about now is what lives in these darkly staining mysterious regions. And as we all know, what we're talking about are highly repetitive or repeat dense regions. These typically involve things called satellite DNAs, which are kind of tandem repeats on steroids that are allowed in these densely staining heterochromatic regions, as well as TE or transposable elements and segmental duplications, which sometimes contain genes. So the work I'm standing before you presenting actually represents the efforts of over 100 people, many giving their time voluntarily as a grassroots effort called the Telomere to Telomere Consortium. We announced the consortium back in 2019. It was kind of a website that Adam Philippi and I had put up with the announcement we completed the X chromosome and said, welcome, who would like to join? And we recruited people who were from first-year graduate school, doing this on the side of their thesis work, all the way to seasoned PIs who were involved with the first Human Genome Project. And what we're celebrating right now is the release of the first finished human reference genome. And not only that, we went through and issued a series of companion papers utilizing this finished reference genome and illustrating some of the biology you can glean from it once you can begin to see these regions. So as in a 1,000-foot view, what we're going to be talking about today is the introduction of nearly 200 million bases that were missing from previous human reference genomes. I know most people in the clinical community are still using HG19, but this is even in reference to HG38. And what we're talking about is also the release of nearly 2,000 gene predictions that were missing from HG38, roughly about 100 that we have evidence to believe that they're protein coding. And throughout it all, we're really celebrating complete maps of acrocentric short arms, centromeric regions, and all segmental duplications. Now, it's important for you to understand before I go forward that what we're also talking about is a very specialized genome in terms of a cell line that we chose to work with. And I want to go ahead and make sure everyone's aware of this, the use of the high titer from mole. We use a cell line called CHM13. And what happens is an early developmental accident. We're really not sure if it happens before or after fertilization, but there's a loss of the maternal genome, as you can see in the cartoon on the bottom. The paternal genome, or the sperm genome, comes in and goes through a round of duplication. Now, why should a T2T consortium focus so much on this very specialized cell line? Well, actually, these very repetitive regions that exist on your maternal and paternal genomes could look very similar in sequence, but could be very different in copy number and structure between the two chromosomes. It's kind of like having a puzzle piece that look very similar, and you're throwing two nearly identical puzzles in the same box and then trying to sort them out and put them back together. Having this effectively haploid genome kind of bypassed that challenge, because now we're looking at two chromosomes, for example, that I'm showing you in blue, where the repeat structure is expected to be identical because they're a duplication of the same chromosome. This is also a story not only of basing it on this effectively haploid cell, but also moving into a new era of long reads, which I know we've been celebrating in the hall next to us. One of the long read platforms, which was incredibly useful to our team, and I want to recognize, was the use of hi-fi data from the PacBio platform. Here, what you're looking at is kind of a multiple pass through a single template, and then you're taking a consensus across these error-prone subreads, understanding these errors presented is not biased, and then all of a sudden you can get an incredibly high-quality circular consensus read or hi-fi data. This is, people sometimes say, like a long Sanger read, since some of us in the audience may be familiar with the quality of Sanger. And what this allows our team to do is actually identify small point mutations or heterogeneity that may exist within the repeats that allow for this overlapping assembly kind of thought process to happen. So that's what I'm trying to show you in the little cartoon over. Another data modality is an amazing accomplishment from the Oxford Nanopore platform. This was something our team was very excited about from the very beginning, and it's actually, I could say, nucleated our team, because Adam Philippi and I were on one of the first papers that were announcing the protocol for ultralong. And so what I'm trying to show you here is that we can now reproducibly reach reads that are 100,000 base pairs extending all the way to a megabase. So no longer do we have to kind of use these 15 to 20 kilobase reads that look at small mutations. You can almost span these highly repetitive regions in their entirety and end on either side. And so really it's this utilization of both highly accurate and extremely long reads, and also giving a nod that these long reads are getting more and more accurate every day, that allowed us to reach our goal. And having these two read databases and cell line is really not enough. You could have thrown those ingredients into any cookbook. I want to take this slide because I don't feel like the students and the early career trainees get enough credit for what they brought to the table. Before we started, no one could actually take these long reads and accurately map them to a 3 megabase tandem repeat with confidence. All of the QC, all of the assembly work, even improving on the long read platforms, that was really a lot of effort that came from this collection of scientists that represent our T2T consortium. So I want to take a moment and credit one particular person in Adam Philippi's group. His name is Sergey Nurk. He was a postdoc at the time. And what he did is he took the hi-fi data, these highly accurate reads, and the nanopore data, and came up with a plan of how we could start to think about assembly strategies to reach complete chromosome assemblies. And the first step was to take the hi-fi data, which was already near perfect, and get it as close to perfect as possible. This was kind of identifying regions that we knew were problematic for these long reads, like homopolymer expansions, or even small dinucleotide repeats, which presented error in the hi-fi data sets. And by identifying that and improving the hi-fi, you could then make something called a string graph, where you're collapsing all of those sequences that are identical into something called nodes, and the edges now represent the connections between those nodes. Instantly, hopefully, your brain can think of how these very long sequences from nanopore can help, where you can use the long reads to kind of allow the pass through the string graph to provide order and orientation. Here I'm showing you kind of the first optics of what this string graph could look like, and what I want your eyes to look at is that all of the individual human chromosomes form their own connected component. Many human chromosomes, like chromosome 11, which I have boxed, came out as a T to T prediction, meaning the ends are there extending into the telomeric sequence, and we have a representation of a human centromeric region just out of Sergei's first approach. There were several regions. They're kind of graying out and also illustrated in boxes that at the time needed serious manual curation and careful assessment by our assembly team. So we had a dedicated team of, I would credit some of the best assembly experts in the field to really lean in and see if they could help resolve some of these assembly predictions. Now, it's not enough to assemble a genome. They could have done it well, but not perfectly. How do you do the other balance of this? How do you start to evaluate quality? And the quality that we issued has a QV score of 73, and what that means is that we estimate one error every 10 million bases. And the way that we did this is also intuitive. Because we had developed a lot of methods now to map sequences accurately to the highly repetitive regions, we could begin to flag any places we saw inconsistency in coverage, any places we saw inconsistency in variant calls, and then we could go through and start to do manual and also this automated curation and polishing. We even had development of new tools to look at these extremely long tandem repeats, these satellite DNAs. One I'd like to call out is VerityMap, or Tandem Cluster, Tandem Mapper. It's gone through a series of name changes, as well as Centrifly, which went through an automated process to see if we can consistently predict the structure of these arrays. And so overall, what we're talking about is kind of the step change from HG38 to CHM13. And as you can see from the pie chart, most of these newly introduced data are falling in something we call SynSat, or the satellites that are typically enriched in the centromeric regions. And what we've issued to the public, and if you'd like more information, of course, ping me, but you can go to the UCSC Genome browser and just pick CHM13, and we have all of our annotated tracks and data presented there now for the community. So this is the joy that I get to step in. I was leading the SynSat working group and also involved with the assembly working group on the early days based on my expertise on satellite DNA. And so having a new and complete reference genome, what can we learn in terms of new genome biology? And questions that one might want to ask is, how can we use this new reference genome to actually improve clinical genomics and variant calling? How can we begin to use it to understand, copy number gene families and segmental duplications, and let alone the centromeric variation that we anticipate finding and these acrocentric short arms? So the first thing I want to go into is our variant team. And the question here we were asking is CHM13, although more complete, is it actually a more accurate reference? Most of the people in this room and most of the people in clinical genomics community are using large cohorts of short read data sets. How much information can you get when you map short read data to HG38, which is the past reference genome, versus CHM13? So that's the question that we're asking. And what we're going to be talking about in the next couple of slides is looking at the 1,000 genomes data set. This was already introduced earlier in a talk where we're looking at roughly 3,000 genomes that represent 26 populations from 5 superpopulations or continental areas. And once again, so I can give you that 1,000-foot view, we're going to be talking about new discovery. We have hundreds of thousands of novel variants per sample that we're identifying in CHM13 that were not present in HG38. We've identified and eliminated roughly 10,000 of spurious variants or false called variants that appear when you map to HG38 versus CHM13. And we're announcing that roughly 12-fold reduction of these false positive calls happen in regions that are deemed to be medically relevant. Okay, so kind of the first mapping, whenever you take these short reads to HG38 versus CHM13, it was very clear to us that we were seeing improvement over a number of alignment characteristics. Here I'm just showing you the mismatch rate, and what I'm wanting you to see is the orange versus blue, and orange being the CHM13 or the newer one, and the blue being the older one. And you can all see that it's showing a lower mismatch rate overall. Additionally, we can begin to quantify this based on the low quality mapping versus the Mendelian violations that we observed mapping to either. And we can also begin to flag these false variant calls where you can see there's an enriched or a larger amount in HG38 versus CHM13. So what's causing this challenge? Most of these highly repetitive regions, if you remember back to my pie chart, were in these centromeric satellites, and short reads don't do a really wonderful job yet in mapping to these particular regions. And so this is where I want to start thinking deeply with this community about why having a T2T finished genome could be useful even outside of those regions that are highly repetitive. We know that the HD38, HD19, HD18, wherever you want to draw the line, is a composite representing a lot of individuals who were represented by back-base clones. And what I'm trying to show you for chromosome 21 is if you look at the top, you can see these shifts of color where you're moving from one back-base clone, which may present a certain ancestry, to another. And they're sometimes next to each other in the genome assembly in a way that presents LD discordancy, or variants that we never observe really in the human population. They're only there because of the architecture of this historic genome project. However, CHM13, because it's giving us an instance of a finished human genome, we can see that it largely represents normal human haplotypes. And we can see this time and time again when we start to survey these LD discordancies, that we don't see the same level of LD discordancy when we search through all of the other 1,000 genomes, including CHM13, and HD38 is clearly an outlier. Another problem that I'm sure you're all understanding is that we have genes that are in HD38, and they might be, in this example that I'm giving you, in 9 copies. However, in CHM13, when we actually look at this same gene family, we're finding a lot more copies of this particular gene. And so if you're now taking your short read data and you're mapping it, those reads are going to find their best location in the genome, and it may not be the true location. It may not have its opportunity to find the best match. And so this is really what we were finding consistently when mapping to HD38 versus CHM13. We found about 203 regions affecting about 8 megabases of sequences and 308 genes. And this includes 48 protein coding genes and medically relevant genes. And we're just seeing over and over again that we're seeing these types of duplication errors that are specific to HD38 that are not in CHM13. Also, when you start to think about something called liftover, these are places where we say, where can we find a match in HD38 that's not present in CHM13? These liftover failures are places where we can't make that one-to-one happen to be impacted in regions that we know are important for clinical genetics and genomic studies as well. So I'm going to take a slight transition from looking at short reads to start talking about what we're learning just by looking at new chromosome architecture in parts of the genome we've never seen before. Now, to me, whenever I'm asked about our project, the first thing I say to family members at the dinner table is, can you believe there are entire human chromosome arms that we hadn't been able to study for 2 decades? You know, because everyone's familiar with the idea of having complete chromosome maps. And so here, having this first complete map of human acrocentric chromosomes, what we're looking at is over 60 megabases of new sequences. And so if we look at chromosome 21, I'll be able to zoom in a little bit more. What our team has been able to do is take these sequences, run our rich annotation and characterization throughout. So now we have deep characterization of the repeats that are there, transposable elements, satellite elements, segmental duplications. And we can also, as I'll go into briefly towards the end of my talk, look into methylation and expression differences and build that type of information-rich map that we've been benefiting from the Q arms for 2 decades. It's not there to the point where we can still have more and more information. Feel the challenge is presented here before you, but still we're at a place where we can start to move forward. And what I want you to pay attention to at the bottom of the slide is all the dark red. What I'm trying to illustrate there is that when we start to do comparisons between the acrocentrics, we're completely in step with what is expected from the literature. These are extremely nearly identical sequences between them. However, that's interesting, right? Understanding how they're similar and how they're different could inform how we think about clinical disorders and conditions that we know are prevalent in the human population. One example would be Robertsonian translocations, where we know that there's a pretty high incidence of 1 in 1,000 births, and it's one of the most common structural variations that we know exist in the human population. But we also know that these events don't always happen in kind of an even playing field across all of the acrocentrics, and we typically see them more commonly between 13, 14, and 21. So now we can go back to these acrocentric maps. And now we can say, okay, yes, they do share similarities. For example, if we were looking at 14, 15, 21, and 22, they have similar satellites, almost like a barcode if you want to look at the colors that way. And they have the same kind of order and organization. And where they're different is really the expansion and contraction of these satellite arrays. Of course, your eyes are saying, but Karen, there's chromosome 13, and that's, of course, very notable to our team as well, that there are these very big differences that exist between the Acrocentrics, such as the human classical satellite that we characterized on 13. But interesting, going back to the Robertsonian translocation story, we did find that there were some similar, highly identical groups that were presented on 13, 14, and 21 alone that were, I'm trying to show here, that were found in different orientations. And so although we're not claiming that this has any role in Robertsonian translocations, I think it's interesting and hypothesis generating to go through and test some of these new features that we're identifying. Also, it was interesting to our team that we saw extreme length variation in these rDNA arrays. You can see, hopefully, that you can see differences from 16 copies to 76 copies. So we went through and we also tried to do copy number variation using different types of assays. Here I'm showing you two digital droplet PCR, as well as looking at this by a quantitative fish. So not only do we have these acrocentric short arm maps, we have all of the sequences and looking at their epigenetic regulation, we can start to move into the maps of human centromeres. Now, as I introduced on my first slide, these are kind of like a kingdom of repeats with tandem repeats and segmental duplications and TEs. And so it was really kind of a fun project to understand the nature of the satellites and their organization in these regions. And the reason why it's interesting and a fun project is because we know that these regions are critical for life. They're critical chromosome structures. They are important for when our chromosomes divide to make sure that they're segregated and separated in each cell division accurately. And of course, if there's an error, this can lead to aneuploidy, and this could lead to all kinds of early development effects, as well as in cancer. And we know that there's also this evidence of proper regulation of these sites. It's not just the sequence alone, it's how they're regulated as we age. We've seen interesting features of heterochromatin being lowered and seeing large senescence features in these regions, as well as in cancers where we're starting to see expression profiles where misregulation is apparent. And for years, these regions have been ignored. We've had wonderful fish-based assays and probes that have been released, and so we are not coming from this at ground zero, and our team tried to connect those dots with the literature as well as we could, but the base level information has been a black box for too long. And so it's been the joy of my career to be able to study these high-resolution maps of centromeric regions. Now, this is a lot of information on one slide, but what I want you to take away from it is that every single human centromere has a dark red block. These are known as alpha satellite, which was introduced at a talk earlier, and had been known for years to be important for where that centromere or the kinetochore forms. If we have rearrangement in these regions, if we put them in artificial chromosomes, it goes on and on. We see the proteins that are important for kinetochores bound to this alpha satellite region. However, you can see there are massive satellites, both in the light blue and the dark purple, known as the classical human satellites, that have been completely missing from map, or we have less information about what they are doing that open new hypotheses for human health and studies. Now, when I was a graduate student at Duke, I actually worked with the first author, Nick Altimos, and what we were trying to do at the time was just understand the basic structure of these satellites. And so I'm showing this kind of a historic moment where we were designing probes and doing elongated chromatin fish to see if we could find these kind of inherent patterns. And it's really cool because we can take those same probes that we worked on way back then and start to look in the context of our current maps. And we can build off of that. So we're moving, what I'm trying to use this slide to say is we're moving not just from my one little one-off as a grad student, but from everybody's one-off that have been building these molecular probes for years. We can begin to see where these probes actually are in the context of a finished genome, and can begin to put even more rich annotation to the type of sequences that we're talking about. We can build a complete catalog of the repeats and the repeat variants, and we can even now go down to the base level and start to look at these DNA binding motifs like the centromere protein Bs, or even other CTCF binding motifs that may be inherent within the centromeric regions. Interesting things that we started to find was that we had the model before that once you're in one of these centromeric arrays, you're kind of, facing forward and putting one foot after the other in a head-to-tail orientation. I think I said that for about 10 years before the T2T. And what we're finding is that there are interesting sites of inversion that could lead to new discoveries for understanding function. Two I'm illustrating here. One is in an array that we know is important for centromere function on chromosome 1. The other is a beta composite array, which is embedded within the chromosome 1 pericentromeric region, where we start to see this checkerboard pattern where the satellites are going forward and then reverse. And this is really striking when you start to look at something like chromosome 9, where we're looking at 27 million bases. I'm just giving you one snapshot of about a megabase. And when we saw this, it took us about a month of proving that this was not an assembly error. It was so striking. And so just understanding why the genome is organized in this way, if it presents secondary structure, if something's going on that's interesting, is this common across other individuals? There's so many questions that come out from finding these new peculiar variations that we hadn't been able to monitor before. Also, in addition to the gene families that we know are expanding in the P and Q arm, as well as the Acrocentrics, we were finding genes that were embedded within centromeric regions. This had been reported before in other studies, and we were lucky to kind of team up with some wonderful members at UC Santa Cruz and also at UConn to go through and marry ProSeq data, which is looking at what the polymerase goes, as well as IsoSeq data, and looking at mapping through GenCode data and libraries to predict these candidates. So finally, I want to kind of pull us into understanding the kinetochore. Centromeres are defined epigenetically. You could put them out in the arm if you wish, if the proteins are located there by neocentromeres. So can we actually use our maps to begin to predict where these kinetochores are sitting? And so what we found in our T2TX paper when we first started was that there are some really interesting things that happen in terms of CpG methylation within human centromeres. And what we were discovering then is that as you start to march across these arrays, there's a very big dip on the order of about 60 kilobases to 100 kilobases in length that happen in methylation. And in these regions, we've also shown, and this is crediting work from Glynis Logsdon's paper on the T2T8, that these are coincident with regions that contain centromere proteins or where the functional kinetochore is being assembled. And beautiful work from Ariel Gershman had shown not only the initial X result, but also that every single centromere in the CHM13 genome and also in terminally differentiated lines that are LCL lines, we see this consistent dip across human centromeres, serving as a diagnostic for where the centromere position is located. And one of the cool features that, see if my can work, yes, wonderful. What I'm trying to show you here is in blue, is an alpha satellite array that contains centromere proteins, so we're going to call it active. And in red, these are also alpha satellite arrays. They're different enough where we can characterize them as array 1, 2, and 3. But there's no reason this array can't contain a kinetochore based on any sequence feature. It's also alpha. And so what we're calling the red are the inactive. Instantly, what we were finding, what I'm hoping you can appreciate by eye, is when you're in an active array, you can see almost like a plateau of CpG methylation. And the second that you're in an inactive array is disorganized. So Ariel tried to quantify this by looking at the CpG methylation across the active versus inactive arrays and also the length of the inactive versus active arrays. And what you can see is that there's a kind of difference. If you're an active array, you're typically bigger, and your methylation profile when taking a summary looks quite different than an inactive array. And this becomes interesting when you start thinking about chromosomes we know that are involved in chromosomal aneuploidies, like chromosome 21, where we note that it's actually kind of looks like an inactive array sometimes in terms of its size, in terms of its amount of methylation. And so these are also another opportunity for hypothesis-driven exploration in chromosome behaviors based on what we're finding epigenetically and genetically. We also now have complete catalogs of segmental duplications. Just as a refresher, these segmental duplications are just any block of DNA that share, that if you were to repeat mask a genome and you were to screen through and find a KB of sequence that's not repeat masked, and then that also KB sequence could fit anywhere else in the genome that's 90% or higher, that is a segmental duplication. And what we found is that in the CHM13 genome, that there's a lot of new segmental duplication. And a lot of these segmental duplications contain genes. That's what I'm trying to show you in this slide, where the red is showing all of the genes that are embedded within segmental duplications that were missing from HG38 and their relative position on their respective chromosomes. And the black are not missing, but yet they were present in HG38 but incorrectly represented, meaning that they were in the wrong orientation or they were incorrectly represented at just the sequence level. And so when we start to study segmental duplications that exist in CHM13, what we found is an increase of the number of large acrocentric segmental duplications. So it's not only a satellite story, there are segmental duplications that are largely embedded between the satellites. And also when we start genotyping individuals from the Simons Diversity Projects, we're going close to 300 short read data sets. What we found is the copy number estimates for each one of these segmental duplications were more similar to what we were observing in CHM13 than in HG38. So this is not a CHM13 better represents cell lines of European ancestry. This is a CHM13 better represents humans in terms of segmental duplication using all of the data that we've had available to us using short reads. Now, the fun thing is when we started to look through these maps, as I shared with you, segmental duplications are regions that don't have repeat massacre data. They don't have any other repeat annotations. There were some pretty big regions with no information. They were not in centromeres. They did not contain segmental duplication. They were just empty spaces that were brand new. And I just think this was such a joy to work with Rachel O'Neill from UConn and her student Savannah Hoyt and the entire repeat team, where they went through and did a whole brand new repeat modeler. And this is also aligning with Arian Smith's team to make sure that we were consistent with repeat massacre and integration in the future. And what they're reporting now is 5.5 megabases of new repeat predictions that were not present in any other human reference genome. And it's cool for many reasons. There's not enough time in this keynote to go into all of the different stories that they were finding, but I thought this was important. And that is a lot of these regions that are missing are highly dynamic. They're known to be highly variable. And a lot of them are found primarily in human or human-specific and not as found in in the non-human primate lineages. And here I'm trying to illustrate that just by the repeat copy number in red and the same type of repeat copy number before in HG38. And also these regions, now we have that really cool feature again, where we have the genetic information and we have the epigenetic information that we can acquire from the long read platforms. And as we start to look at CpG methylation over these novel repeats, we're also finding interesting regions of regulation in terms of finding dips and valleys and places that would look otherwise identical or uniform over these tandem repeats. And so this has been something that I think will be interesting as we start to move into other cell lines to see if this type of variation at the epigenetic level can be recaptured. So I used to hate this image because whenever I was talking about the Human Genome Project not being completed, my colleagues would always say, well, it's like climbing Everest. It's just a big challenge. And I'd be like, no, we're in the jungle. You know, we're all these things that we've never known before. We're opening it up for the first time. You know, the satellites are important. But now I've kind of started ending my talks with this picture because I think that what we have done is this Herculean effort. It took a lot of training, it took a lot of expertise for our team to get here, but it changed the way we view outward once we got to the top. And I think all of our team is shoulder and shoulder in announcing that we just recognize that what we've done is simply not enough. One genome is not enough. We have a really big technological barrier in front of us. We need to start moving in a way where we're not depending on a specialized cell line anymore. And so this has been a lot of work since the release of our paper, and I want to credit Adam Phillippe's team in particular in the release of a software called Virco. And what Virco is trying to do is automate some of the processes that we were doing for the CHM13 work, where we're utilizing the two long read platforms I introduced at the beginning of my talk. These long, somewhat error-prone reads from Oxford Nanopore, but can extend hundreds to a megabase in length, and also using a long, accurate read, such as Hi-Fi, but we're also interested in exploring any kind of duplex read that was from ONT to start to build these initial string graphs. So it's going through in more of an automated procedure of identifying these long overlaps between highly accurate reads and threading through with ultra-long, and then we're using Hi-C data or parent data to phase. And we're making tremendous progress on T2T diploid. In fact, so much that we're starting to ramp up and look at panels of T2T genomes right now. So I wanted to at least include this at the end of my slide to put a note toward the future of where we're going. I think we're very quickly moving to a place where diploid genomes at the same level of completion that we're seeing for CHM13 are possible. And that's why it's important to transition into this slide, because having one genome is not enough. And even at the beginning, when I was making my argument about the incomplete nature of our human reference genome, what I didn't spend time on is that having one genome of European ancestry is not enough. It introduced biases, it introduces questions of inequity, and so what has been a call to action from NHGRI is really to see if we could turn our technology not only to trying to resolve one human diploid, but can we do this on a production scale for hundreds of human reference genomes? And so that's where I switch hats in front of you as the director of our production center for the Human Pangenome Reference Consortium, where it really is a goal or a call to action to improve complete genome representation of a collection of individuals who represent our species. In terms, right now we're aiming for at least 350 diverse individuals. This was based on small mappable SNP-based estimates from 1,000 genomes to represent 1% sequence variance. We understand we'll likely need more in order to capture the larger structural variants in complex regions. But in doing so, this is a technical assignment in kind of reaching a complete and comprehensive map of genome variation. In addition to that, it really emphasizes what in the world is the community going to do with hundreds and hundreds of complete genomes. So we have to really turn the world on its head and start asking about the data structure. How do you begin to work with a nonlinear reference genome? How do you begin to adopt a whole ecosystem of something that we call pangenomic tools and make them accessible to the community? So in doing this, we're in our year 3, so we've put a tremendous amount of effort. We recently released about 47 of our reference genomes, or 94 of these phased haplotypes. Here I'm just breaking down the individuals that were part of that release. And our goal is, like I said before, is to represent genetic diversity as well as biogeographical diversity. So once we have these genomes, which I can talk about in terms of our long-read platform and our coverage estimates, if anyone's interested afterwards, but is to release a T2T genome. So everything's kind of in mode of how can we get to that production scale to reach what I just presented the advantages are in the CHM13, but do it for individuals of diverse ancestry available to us. And how can we then take these, what I'm showing you here, the maternal and paternally phased genomes, maybe in the dark gray and black, and take these maternal and paternally phased assemblies and convert it into a graph or a string graph. Where here, what I'm trying to show is the regions or the shared haplotypes between individuals can be collapsed as a node, and regions that they differ, maybe it's an inversion or insertion, can be represented as variations in this graph. This is big science at its best and involves a multi-center reference production across many different regions, really crediting a tremendous amount of work once again to generate hi-fi data where we have an estimate of about 35x coverage. We need to have at least 30x coverage of ultra-long, not total coverage, that's ultra-long. That means we need at least 30x span of the genome of 100 kb plus reads. And we're also using OmniSeq from the Dovetail platform to help us with phasing. And then all of these data types are going through on a cloud base, crediting our collaborators at Amazon to bring them onto the public database, as well as Anvil and Terra to create workflows. And we go through automated assembly and quality assessment at Santa Cruz. And these are really spectacular assemblies, even though they're not yet T2T. What we can see here is that if you have the dotted line that I've provided, which is the T2T genome at 3.055 gigabases, most of our assemblies are meeting that. Where you see the dip is where we have an expectation for a dip, because that's representing the paternal or male component of that particular genome where we expect to have a Y versus an X, and that is the amount we would expect to see dipping. Also, these are extremely high quality, as you're looking at the scatter plot at the bottom, where we're seeing consistently high quality in the range of 50 to 60 QV scores. So I'm going to end my talk with always announcing kind of not dirty laundry, but technical challenges and places that we're really trying to lean in right now. As I already introduced, we are making progress towards T2T, but we're definitely not there yet. This is something that we're all trying to work toward automation so that reference genomes can be of equal quality and variant calls between these genomes can be trusted. In addition, once we have these genomes, how do we begin to model these highly variable regions between them in a way that's useful to the community? And how do we begin to call variants in these regions or build the protocols that are necessary for outreach and engagement with communities that are interested in flagging new associations with disease? So I definitely want to emphasize that these assemblies, in my own biased opinion, is going to revolutionize what we're going to do over the next 10 years. You might hear my talk today, but it's my hope that in 10 years will be very boring and an assumed thing that the genomes that you're working with will be of this quality. And by doing so, we're hoping that that will illuminate new questions and studies that have range in healthcare. Also helps us to understand genome diversity as we start to share methods and ideas with other communities of biomedical research and interests such as the mouse and rat and fly and non-human primate communities to improve reference genomes kind of outside of humans as well. And also I think there's a new opportunity to study cell biology in a comprehensive way where we're not only aiming at parts of the genome that we can measure confidently with short reads, but aiming comprehensively for the good of the patients and for the good of science, to find discoveries that maybe we couldn't see before by a technical limitation. And with that, I want to glorify and be very grateful to the amazing people that I am presenting on behalf of. It has been the honor of my life to be part of T2T. These are amazing people who have tirelessly put in, even throughout the pandemic, effort to be able to present the work that I'm showing you today. And also I want to acknowledge the Human Pangenome Reference Consortium, which is a tremendous effort as well to ensure that we have a more equitable and more better representation of our species in the next go-around for human references. And this is really crediting the intro that hopefully no one throws tomatoes at me when I say we're never complete, but I do feel like we are moving toward a better idea of what completion means in terms of being finished and in terms of representation. So thank you for your attention.
Please go ahead with, we do have time for questions.
Dr. Miga, fantastic talk. So I'm Patrick Gonzalez, KU Medical Center, also a genome nerd. I have been following this work and I've been waiting for it for years.
Oh, good.
So I'm sure that I can speak for many in this room that have been eagerly waiting for this data to come out. So one question that I have, I think that most, if not nearly all, genetic testing laboratories in the United States and probably worldwide are still using HG19 as the reference genome. What is your recommendation, if you have one, for labs that are wanting to make a change to a new reference genome? Should they go with, GRCH38 or wait for the T2T to have more definitive kind of pan-genomic references for clinical genomic testing?
I think our official stance in working with our colleagues at in CBI and trying to think through how to advise the public, when this question is presented, is that we see HG38 and HG19 to some extent as kind of a companion or parallel reference assembly with CHM13. We never want to replace a reference genome of the past with ours because it has so much historic legacy data that's useful to a researcher. Personally, if I can speak outside of this kind of mantra of where we are with the T2T consortium, I've been advising researchers that if they're interested, just do exactly what we did for the 1,000 genomes test. Just run a sample between the two genomes and assess benefit for your question. Now, there'll be many researchers that are just wanting to design assays for a particular gene, and that gene's not changed between the two references. And so it's hard to ask such researchers to switch over for that. But if you are doing comprehensive genomic screens, and some of the information that you could reach now could be within these 200 million bases, I could imagine it would be advantageous for clinical care outcomes and new discoveries to do that primary test. And I also envision that we are moving so fast with the pangenome and what we want to build for tooling for the pangenome, that there is a real initiative for me to also think about that big leap, that big Herculean task of outreach to make people switch from HG19 to something should be focused on the pangenome, because CHM13, HG19, HG38, they're all paths in that pangenome, right? So it's more or less added value. You're not losing value at that point.
Thank you very much for the answer to that. That's been fantastic.
Thank you for the question.
So I kind of have a companion question to that. In thinking about people upgrading, have you thought about using productized assays and things that are out there to see what the actual difference is for genes that people are targeting for their clinical assays to make this easier? Because not everybody has a bioinformatician who can just kind of do this.
Right. I think that for the CHM13 genome, we pretty much just released it, and all the genome geeks were like, go to town, folks. We didn't think about, how can we begin to bridge this into a more formalized diagnostic assay? How can we do proof of principle? How can we make this more of a realistic bridge to folks who want to use it? And I think that's in the spirit of your question. I think where perhaps that impact could be, once again, is with the Pangenome project, where now you have CHM13 as one of the paths of, the collection of these more complete genomes. And maybe by engagement with consortium-based efforts, which already have, a huge amount of clinically important short read data sets, to create that bridge to see if we can identify areas that could develop a new assay, or work with groups that are focused on a particular gene, but they've only been looking at one instance of that gene. If there's an opportunity to kind of bridge into new assays that bring in that lens of diversity into their study. I feel like every single gene in the genome now has its own story versus CHM13, which was just one. So it does give a broader, scope to engagement, which is exciting. And I think that that's where we'll be in the next 2 years, to start making those really impactful contributions and bridges.
Great, thank you.
Hi, thanks so much for your talk. It's really exciting, and you're in really great company of other genome geeks here, so
Oh, good.
just know that. So when I was listening to your talk, I'm thinking about conventional chromosome analysis. We're looking through the microscope. A lot of times we see those variable regions that you talked about, like chromosomes 1, 9, 16, and we will actually track those chromosomes by the size of their centromere. I'm kind of wondering if it's in scope for the T2T project to kind of quantify the number of repeats in those regions in a normal range, kind of like we do trinucleotide repeats, or if that's something that we should really be thinking about kind of outside of that project.
So for estimates on the largest classical satellite arrays, it's always been tough for me personally to think about how to do that using short read data, because you're looking over two diploids. They can have one extremely long array, one very short array, and you're kind of taking the average because of the lack of ability to phase. Now, this is an opportunity, I think, for the human pangenome release, where we now have those, 94 assemblies, which present a tremendous amount of new satellite DNA. So we've gone through and analyzed those. I'm not presenting the data, but I'm happy to meet with you after, where we're finding, if I draw the line, this is where CHM13 presents HSAT2 or 3. We see that beautiful clustering of the same percentage of the genome. And when we start to plot the array prediction sizes that we now can phase between maternal and paternal, then we say, okay, show us the biggest arrays, for example, on chromosome 9. We have been taking cytogenetic studies of each one of these cell lines as we go. And so we can go back into the Coriell cytogenetic files, which are already marked as having a 9H, het expansion, and find that kind of crosstalk between what we're seeing at the assembly level with that. I do think that we're going to need bigger numbers, but I think as far as the assessment of the size variation, it may require this level of assembly so that you can get that confidence that you're looking at a big array on one chromosome and a small array on the other, or two big arrays. I think that that's something that will probably require a little bit more finesse to make some concrete conclusions.
Thanks so much.
Hi, I'm Patty Myron from UMass Memorial Medical Center, and that was just a wonderful, wonderful talk.
Thank you.
My question is probably a little bit related to Beth's, and first back to your comment about looking at trisomy 21 in non-disjunction. And I think about 21 as being common because the chromosome is so small, so it's viable. And I'm wondering if you've looked at chromosome 16, which is, so common in non-disjunction, and if you've looked at the centromere for that chromosome.
We have. We don't see anything that I would say is out of the ordinary, like chromosome 21, or I think this has come up, and I think it's quite interesting. I think when we start to look outside of CHM13, that may be a better point to put a pin on that to come back and talk to you. Because it might be that 16 is highly variable in the human population in terms of its composition and array length, and we're just looking at one instance of it with CHM13. And so this has been brought to my attention that there are these very interesting chromosomes that present interesting non-disjunction features, and some of them, like 16, have these big classical satellite blocks in the pericentromeric region as well. And so I think that there's really an opportunity here to go into some of these models and explore sequence features and also epigenetic features. And if you don't mind me taking liberty to build on that statement, epigenetics features, it was when we were looking at the X chromosome. That's because in males, X is haploid, so we could kind of turn the crank and start looking at some of these X arrays. And of course, we found genetic variation, so that was cool. But what was striking to me was the epigenetic variation. So the centromeres sometimes will be on the P arm proximal, sometimes will be center, sometimes be Q arm proximal. And what we were questioning is, is this an ascertainment bias? In other words, are these, just because X, you can have such flexibility in aneuploidy, maybe it allows the centromere to have this type of flexibility. And maybe if we looked across the epigenetic profile or variation of all the different centromeres in addition to the sequence, we might find that some of this epigenetic variation is more inherent in some centromeric regions in the human population than others. And that may have a consequence because at the end of the day, it really is this site of kinetochore that's the most impactful, we believe, for functional realms in these regions.
Yeah. Thank you very much.
Thank you.
Hi, I'm Bryn Levy, Columbia University. Just an amazing talk. It's just so refreshing to see this type of data. Again, as cytogeneticists for many years, we've seen the polymorphisms in the heterochromatic regions, and Linda Bourne also spoke this morning, I think you alluded to that, with respect to the effects of these polymorphisms that we're seeing, by using FISH probes. So one of the things we've known is that you can actually see centromeric regions, or probably even heterochromatic regions, moving to different chromosomes. So you're almost going to get chimeric kind of sequences. So number one, have you seen that? And is there any thought to how these kind of chimeric chromosomes that represent, evolutionary jumping around, how you would represent those types of maps? And I'm sure they may be population specific.
We're just now starting to scratch the surface of that. I'm sharing your enthusiasm based on this published literature looking at cytogenetic mapping and characterization, but not at the sequence level yet. Where we saw the first instance of kind of the emergence of these satellite arrays was on chromosome 1, where if you're looking between the centromeric region and this massive classical human satellite repeat, in some individuals we're finding a 0.5 megabase of satellite, and in others it's not there. So there are some really big structural variants in terms of just satellite composition, the presence or absence of massive arrays that is in its own self a huge and a very interesting project to undergo. I think that the idea that, for example, you have the gain of one satellite and the emergence of another, could also play into our ideas of how the centromere functions. And I would also like to even put out there that one of the most interesting things that I found before the T2T Consortium was looking at these large linkage blocks that we know span each centromeric. So in other words, centromeric regions, as we're all aware, because of their presumably darkly staining heterochromatin, are not influenced by meiotic recombination. So they typically, the sequences within these regions are inherited as blocks or haplotypic blocks or centromere haps, as we sometimes call them. And what we found is when we start to look at the sequences on either side of these linkage blocks that are mappable, that we can start to use phylogenetic tricks to classify, that some of these blocks look really, really old and could be archaic integration. And so it could be that some of these events that present these satellites in a very interesting way could be from Neanderthal or could be from Denisovan. It's just, I think that there are a question there as well of kind of looking in the context of ancestry and also in the terms of SynHap blocks too.
Yeah, thanks.
Thank you.